Cancer. One of the incurable diseases of our world, cancer continues to take the lives of millions. But what prevents it on a genetic level? Well, let's zoom into one of the most important genes known to prevent cancer, MSH6. MSH6 is a member of a set of genes called mismatch repair genes, which are a type of DNA repair enzyme, which is a subclass of tumor suppressor genes. The MSH6 gene provides instructions for making a protein that plays an essential role in repairing DNA. This protein helps fix errors that are made when DNA is replicated in preparation for cell division. Everyone has 2 copies of the MSH6 gene, which is located on chromosome 2 that we randomly inherit from each of our parents. Thanks, Mom and Dad. Now, the MSH6 protein joins with another protein called MSH2 to form a dimer. No, not the coin, a dimer. A 2-protein complex that identifies locations on the DNA where errors have been made during replication, thus preventing the division of abnormal cells. But what happens when the MSH6 genes become mutated? Well, let's back it up a little. How do MSH6 genes become mutated? MSH6 can be transfigured through germline mutations, which are passed from parent to offspring. These are changes in the body's reproductive cells that become incorporated into the DNA of every cell in the body of the offspring. The mutated version of MSH6 leads to 2 major conditions: constitutional mismatch repair deficiency syndrome and Lynch syndrome. Let's start with CMMRD. There's over 15 variants of MSH6 associated with this condition, but they all have one thing in common. There's mutations in both copies of the MSH6 gene. The variants result in near or complete loss of the MSH6 protein production. A shortage of this protein eliminates mismatch repair activity and prevents the proper repair of DNA replication errors. These errors accumulate as abnormal cells continue to divide, eventually disrupting other genes involved in important cellular processes such as controlling cell growth and division. If cell growth is uncontrolled, it can lead to childhood cancer. Now, Lynch syndrome is caused by a mutation in only one copy of the MSH6 gene. The variants involved in this condition lead to the production of an abnormally short, non-functional MSH6 protein or a partially active version of the protein. Since there is some functional protein produced from the normal copy of the gene, mismatch repair activity in Lynch syndrome is reduced but not absent, as it is in CMMRD syndrome. This difference in DNA repair activity levels probably explains why cancers in Lynch syndrome usually develop in adulthood, while those in CMMRD syndrome often affect children. But either way, a decrease in functional MSH6 protein leads to an increase in unrepaired DNA errors during cell division, once again increasing the risk of tumor formation. The most common cancers associated with mutated MSH6 are colorectal cancer and endometrial cancer, but other cancers are also associated with this mutation, including ovarian cancer, stomach cancer, small intestine cancer, urinary tract cancer, pancreatic and hepatobiliary tract cancer, brain cancer, and sebaceous carcinoma. Whew, well, there you have it. Our MSH6 genes do more for us than most of us know, so when you get a chance, thank these small but mighty detectives saving you from a possible case of cancer.